In this lab, we've been investigating the PTC taster gene. So far, we've learned that about 75% of the population can taste this, and when they do, it's very bitter, and the other 25% cannot. We've also seen that in 2001, the gene that controls this trait was sequenced and published. So in a prior activity, we collected the gene sequences for both the taster and the non-taster alleles. As you can see, the two sequences look identical, but there are differences. So in this part of the lab, we're going to use the bioserver's website to help us identify the differences between the two sequences. So when you enter the bioserver's page, you'll be given three choices: sequence server, allele server, and simulation server. For our purposes, we're going to use the sequence server, and we're going to enter into the sequence server. When we do so, we're presented with this window. One of the first things we'll need to do is to populate the window with our sequences. So up here we see create sequence. We're going to create a sequence. The first sequence we're going to put in is the taster gene. We've acquired this from a previous lab. One thing to note that there might be lots of information from that previous part of the lab, but we only want the DNA. So just copy the DNA from that. We're going to paste it into this window here and hit OK. And when we do so, we'll see that the taster gene now populates the work area. We're going to do the same thing for the non-taster gene. We're going to name it non-taster and then go to what we had collected previously, the non-taster, just the DNA, paste it into here and hit OK. And we'll see then that we have the non-taster as well. So now we're ready to compare the two genes. To do so, we're going to select the genes we want to compare. So make sure that they have this blue check mark, and then we're going to hit the compare button. And now it's showing the two genes lined up. Some information at the top here that's important. The alignments are in blocks of 25 nucleotides. So notice 25, 50, 75, and so on. Mismatches are colored in yellow. Now, the gene is 1,002 base pairs long, but it's only showing us 500. So we're going to change this number at this point to 1,100 and then redraw. And now we can see our first mismatch here on the line that reads 150, and there's another one down here at 800 and another one at the line 900. So we want to know where these mismatches occur. So we're looking at the first one here at 150. What we notice is that this G at the end here is the 150 position. But if we count backwards, we can find exactly where the mismatch occurs. So 50, 49, 48, 47, 46, 145 is where that first mismatch occurs. And we can also tell what the difference is. The taster gene has a C nucleotide, and the non-taster gene has a G nucleotide at that position. Further down, we see at 800, this line here. Now, notice the mismatch is toward the other end. So an easier way to do this would be to count forward. So this T is 775. So counting forward, 76, 77, 78, 79, 80, 81, 82, 83, 84, 85. 785 is the position for the second one, and the C, taster has a C, and the non-taster has a G. And then further down again, this is at 886. The taster has a G, and the non-taster has an A. So with that, our analysis of the two genes is done. So in this video, we saw how to submit DNA sequences into the bioserver sequence server, and then analyze the two sequences side by side to identify nucleotide differences. In the next part of this lab, we will add more sequences and investigate other aspects of the taster gene.